I do want to do this. Like, I do all of 4 and 7, and you do all of 5 and 6, or switch off. So,
Well, you'll do the first topic and then I go.
okay, perfect.
Wait, is it recording? I didn't hear that you were recording. Okay, yeah.
Okay, yeah.
Okay. I'm Rosella. I'm Liv, and this is exam 2 review. And we're gonna just go through like the different topics, and then we'll have time for questions later. But you can also ask questions throughout. So we'll just get started. Okay, so I'm gonna start with topic 4. Wait, Rosella, how do I go to the next slide when it like looks like that and it's not on here?
You can just
click. Oh, okay, got it. And then, yeah. Okay, so I'm going to start with topic 4, which is mechanisms of gene expression regulation. So to kind of give you a little overview to start, things go a little differently in eukaryotes and prokaryotes. So in both eukaryotes and prokaryotes, they use transcription factors. RNA can't bind to DNA on its own. But eukaryotes use basal transcription factors and prokaryotes use sigma factors. In both regulatory transcription factors control gene expression. They are gene specific, and a positive regulatory transcription factor will upregulate the transcription of a gene, and a negatively regulatory transcription factor will downregulate transcription. And I already mentioned that, but RNA polymerase cannot bind on its own because it's not sequence specific. It has to be able to go along DNA strand without specificity. In prokaryotes, sigma 70 is the transcription factor that helps RNA polymerase to bind. It recognizes a consensus sequence, which is just basically the most common sequence of nucleotides along a specific sequence. So it's not going to be the same for every single organism, every single gene, but it's the most commonly recognized nucleotide at each position that sigma 70 can recognize and bind to, and then RNA polymerase will bind to sigma 70. Going into more detail on consensus sequences, they're determined by aligning the target binding site, then seeing which nucleotides are most common at each position. So, for example, for nucleotide position 7, right here, you can see that there's AA, CAA, GAA. The most common nucleotide there is A. So for the consensus sequence, you would pull an A for position 7. And then for position 8, it's mostly C's. So the position 7 nucleotide would be C. So then for each different organism, go down at each individual nucleotide position, and the one that's most common is the one that you're going to find in the consensus sequence that the transcription factor can recognize. So in prokaryotes, an operon is a series of genes that are controlled by the same promoter, and they are transcribed at the same time. So one strand of RNA has more than one protein coding region. So for example, in one operon, there's multiple structural genes that will all be on the same mRNA strand that can be translated from the same strand. So the main operon that we talked about is the trip operon. So to kind of get into important terms that go with the trip operon, the promoter is where RNA polymerase binds. The operator is where repressors bind. It's inducible, usually off, but can be turned on. And it's repressible, which means usually on or turned off. And for example, that is the trip operon, usually on, turned off. So what is the trip operon? It's an operon in E. coli that codes for metabolic proteins needed to make tryptophan. And when there's a lot of tryptophan around, the trip operon will not be transcribed. So here's a little cartoon kind of help you remember that the car can't get by to transcribe trip because there's too much trip already in the way.
Yeah, so it goes from metabolic. Goes from metabolic, protein to tryptophan, and doesn't do it if there's something that's been.
Exactly. So at low trip levels in the cell, there's no access to tryptophan, so there's need for more tryptophan. So then tryptophan genes will be on and transcribed. At high tryptophan levels, on the other hand, there's abundant access to tryptophan, so there isn't really a need to transcribe more. So then those tryptophan genes will be turned off and not transcribed. So the way that that works is tryptophan binds a repressor that is transcribed by TRPR. And then this repressor, when bound to TRP, will bind to DNA and will block the place where sigma 70 would bind. And then if sigma 70 can't bind, RNA polymerase can't bind, and then the TRP operon will not be expressed, TRP will not be transcribed. All right, so then we have attenuation, which is early transcription termination. This is also dependent on TRP tRNA levels. So RNA can base pair with itself. And these occur in the form of hairpins, which are small, and stem loops, which are larger. And these sequences are before the TRP genes where attenuation will occur. And there are 3 hairpins / loops that can occur where there is affinity between the 2 strands and that or complementarity. And those are the first and second regions, second and third and fourth. All right, so again, at high levels of tryptophan, tryptophan will be bound to the repressor will be bound to the operator, transcription factor can't bind, RNA polymerase can't bind, and there will be no transcription. Whereas at low levels of tryptophan, tryptophan is not bound to the repressor will not bind to the operator, and full transcription and translation can take place. Okay, so the way that this mechanism works, so at the 1 and 2 regions, there will be a pause as the ribosome catches up to RNA polymerase during translation. So as the mRNA is being transcribed, the ribosome is translating the mRNA strand. So the ribosome gets stuck in region 1. If no TRP is available to add to chain, the second and third regions form a loop, and there will be no attenuation. So again, if there's no TRP available, there's no attenuation, which means that transcription will proceed. So the 2-3 hairpin is the continue signal. Transcription and translation continues, and a full mRNA transcript is produced by the operon. When there are low to high levels of tryptophan, just to refresh your memory, tryptophan will not be bound to the repressor, so then the repressor will not be bound to the operator, and there will be transcription and translation. But, right, we're going to post these slides after you get an email, so if you need more specific or a different, to have these concepts explained in a different way, you can always watch these videos. Just going to click out of here.
The TRP operon in E. coli contains 5 structural.
All right, so for a quick summary, at high tryptophan levels, the repressor will be bound. There will be no transcription at all. At low tryptophan levels, transcription will be started. 2 and 3 hairpin form, which means transcription will continue, as well as translation. But when there's low to high tryptophan levels, transcription is started, but the 3 and 4 hairpin will form, which causes attenuation. So I think there might be a slide missing here, but 1 and 2 hairpin is just a pause. 2 and 3 means continue transcription, and 3 and 4 will call for attenuation, and that will occur when there are high amounts of tryptophan in the cell. Okay, so going into non-coding RNAs, these are RNA molecules that do not code for proteins, so think like not mRNA. They have different roles and functions in the cell, such as reaction catalysis and gene expression regulation. For example, we have rRNA or ribosomal RNA, which you're probably familiar with, catalytic parts of ribosomes, aka ribozymes, and these catalyze the peptide bond formation when you are translating mRNA into a protein, connecting individual amino acids together. We have tRNA, which is transfer RNA, which brings amino acids to the ribosome during translation. And they bring the amino acids into base pair with the mRNA strand, and then the ribosome will connect those together. Small nuclear RNAs are the catalytic part of spliceosomes. Long non-coding RNAs are about 17,000 base pairs, and they're involved in X chromosome inactivation. And then you have CRISPR RNA and SRP, ribonucleoprotein used in protein translocation. Okay, so getting into RNA interference, one of the ways that non-coding RNAs can function.
So are there gonna be exam questions about the big that were on the last slide?
I don't think so, right? Like specific, like especially not like small nuclear long non-coding. Yeah, I think these are just examples. I wouldn't worry too much about the super specific information. Maybe just have an idea of like a few examples, rRNA, tRNA, those are the big ones.
Although small nucleotide ones and those they were mentioned that in the questions. I also think the CRISPR role you mentioned. That would help.
Yeah, I mean, I can't say for sure, but I mean, it doesn't hurt to put it on your sheet if you have space, but these specific details aren't the most important thing for you to focus on for the exam, I'd say.
This is important, right?
Yes. Yeah. Okay, so going into RNA interference, this is a eukaryotic mechanism of gene expression regulation, and there are similar mechanisms in prokaryotes. It is post-transcriptional control of mRNA via suppression, so after the DNA has been transcribed into mRNA. Some of the important terms here are RISC, RNA interference machine, RNA-induced silencing complex, which will be the protein that splits apart base paired miRNAs, microRNAs or miRNA, and short interfering RNA or siRNA. And we'll get into more detail about the mechanisms for each of these. All right, so going further into RNA-induced silencing complex or RISC. It is an RNAi machine complex with multiple proteins involved. The general mechanism is that RNA binds to RISC, and RNA is used as a guiding strand targeting specific mRNAs. So the RNA we're talking about that is targeting specific mRNAs is miRNA, and it recognizes specific consensus sequences on the mRNA by base pairing. And then by recognizing the base pair sequences on mRNA, it can regulate translation or degradation of those mRNA strands that the miRNA recognizes. So two pathways that this takes place is through miRNA and siRNA. So going a little bit into miRNA or microRNA. This is a post-transcriptional regulator, as I mentioned before. It's a guide strand for RISC, and regulates mRNA stability and localization by making those complementary base pairs. It's endogenously expressed, which means that it is encoded for within the genome, and it is expressed in the nucleus and has to be exported from the nucleus. It's not incredibly prevalent in humans, thought to regulate less than 60% of humans' protein coding genes. Okay, so for the specific pathway, starts out as pre-miRNA, then it is processed by Drosha in the nucleus, and then it is exported into the cytosol by exportin, after being processed by Drosha. Actually, I made the scissors. And then once it's in the cytoplasm, second cuts are made by Dicer, and then we have these miRNA duplexes that are unwound or pulled apart by RISC, which we talked a little bit about before. And then one of the strands will be degraded, see on the left there, and then the other strand, which is bound to RISC, will be used as a guide strand to recognize mRNA and cause it to be degraded, or whatever the downstream effects are meant to be. Okay, so once we have the guide strands with RISC, base pairs complementarily with target mRNA, and the end result is translational suppression or sequestration to P bodies, where we're not really sure what happens to it after that, but just know that the mRNA recognizing mRNA will cause it either translational suppression or sequestration to P bodies. In humans, mRNA does not exactly base pair with target mRNA, which allows for one mRNA to potentially regulate multiple mRNAs. So it can basically recognize multiple different sequences with slight variations. But if it's an exact match, then Argonaut, which is the catalytic component of RISC, will be activated and direct mRNA degradation. Okay, and then we have small interfering RNA, or siRNA, which is exogenously expressed, which means it's usually brought in from somewhere outside the cell, such as from a virus. It is also a post-transcriptional regulator, similar to miRNA, also a guide strand for RISC, also regulates mRNA stability and degradation, and it's usually a small 20 to 25 base pair of double-stranded RNA. Key difference from miRNA is that it can protect organisms against foreign invaders that contain dsRNA or viral infections, because they can recognize those sequences. And also different from miRNA, it's usually an exact base pairing match. So remember, for miRNA, it's not necessarily, or it's usually not the same. So basic pathway for siRNA starts in the cytoplasm. Since it's exogenous, it doesn't need to be exported from the nucleus like miRNA does. dsRNA or shRNA is cleaved by Dicer. So that's those original strands of RNA. And then we have the siRNA duplex. RISC unwinds the duplex and picks a guide strand with help of Argonaut, which is part of the complex. And then the RISC siRNA complex forms. And then as I mentioned before, there's an exact base pairing match between siRNA and the target mRNA, which leads to a cleavage of mRNA by Argonaut. And this leads to mRNA degradation or silencing. And then as a final summary to give you an idea of the main differences between the two types of RNA, non-coding RNA. mRNA is endogenous, produced within the cell, while siRNA is exogenous, comes from outside the cell. mRNA has Joe-strip processing, then dicer processing because it has to be processed in the nucleus before leaving. Whereas siRNA only has dicer processing because it's immediately in the cytoplasm. mRNA is usually not an exact base pairing match, while siRNA is usually an exact base pairing match. Translational suppression or P-body localization for silencing or degradation with miRNA, whereas for siRNA, usually there's just immediate activation of Argonaut, which leads to cleavage slash degradation. Okay, so now we're gonna go through, there's the chair over there. Okay, we're gonna go through cell signaling and signal transduction. Okay, so all organisms and cell types have the ability to receive signals, and cells need to receive signals from their extracellular environments in order to appropriately respond to changes in their environment. So like changes in available nutrients or amino acids. And then cells can receive multiple signals at the same time and express multiple different receptors to be able to receive these different signals. Okay, so chemistry of signaling molecules. Signals are very small molecules or proteins, and they can vary in size, shape, and chemistry. Because of this, some signals can pass through the cell membrane and some can't. The signals that can't pass through the cell membrane are too large or too hydrophilic to pass through the hydrophobic phospholipid tails. So the cell membrane is made up of the phospholipid bilayer, and it has like the hydrophilic phospholipid heads and then the phospholipid tails that are hydrophobic. So if the signal is too hydrophilic, then it can't pass through the hydrophobic parts. And then signals that can pass through are small enough to pass through the membrane, but are also hydrophobic enough to pass through the phospholipid tails, but not too hydrophobic that it's repelled by the hydrophilic heads. And over here you can see the molecules or the signals that can't pass through the cell membrane usually bind to an extracellular a cell surface receptor, which then causes the signal to be transduced in the cell. And then for signals that can pass through, they will bind a receptor inside the cell. And then the signal is transduced that way. Something important to note is that the physical signal itself is not what gets passed along in a cell transduction pathway, but it's just the information from the signal that is transduced through conformational changes after the signal binds a receptor. So over here you can see the signal molecule binds the cell surface receptor, which then causes a conformational change that activates different downstream proteins in the signal transduction pathway, which then eventually leads to whatever appropriate response. And there are two different outcomes of signal transduction. You can either have a fast response or a slow response. In the fast response, what occurs is that an already existing protein changes its function. And this is a fast change because the cell doesn't need to produce anything new. And in the slow response, gene expression is changed. This is a slow response because you have to go through transcription and translation in order to lead to the desired outcome. And then these responses are not mutually exclusive. A signal can cause both a fast response and a slow response. In a signal transduction pathway, you can have both amplification and crosstalk. In amplification, what happens is that components of the pathway can activate multiple downstream targets, amplifying the signal. So you can have a larger and faster cell response. And then in crosstalk, it's a process where components of one pathway can inhibit or activate components of another pathway, which just allows for more regulation. So the crosstalk is like the horizontal arrows between the pathways. Any questions? Okay, so now we're going to get into different examples of eukaryotic signal transduction pathways. And the first one is the RTK RAS-MAPK pathway. The purpose of this pathway is to signal for cell division and differentiation. And the type of receptor used is a receptor tyrosine kinase, or RTK. So the steps to how an RTK receptor works is that, number one, the signal binds the lone RTKs, which cause them to dimerize, and this dimerization activates the RTK's kinase activity through a conformational shift. And because both RTKs in the dimer are now activated as a kinase, they can cross-phosphorylate each other. So each dimer phosphorylates each other, causing bindings, tyrosine phosphatase binding sites. Sorry. And then these phosphorylated tyrosines create binding sites for downstream proteins in whatever pathway needs to occur with this RTK. And then the proteins that bind the phosphotyrosines have SH2 domains, which we'll get into again a little later. Any questions? Okay, so just some practice with RTKs. Will the RTK still dimerize if the tyrosine kinase domains are mutated such that the cross-phosphorylation cannot occur? Yes, because the RTKs dimerize before cross-phosphorylation. And then two, can the RTK still phosphorylate if the signal cannot dimerize? No, it can't, because if the signal can't dimerize, then the RTKs aren't activated, and then their kinase activity is not activated, so they can't cross-phosphorylate. Okay, so now we're going to talk about a specific protein that will bind to these tyrosine kinase domains, and that is GRB2, which is also called the adapter protein. So this GRB2 that binds the phosphotyrosine domain has a SH2 domain, and an SH2 domain is just the protein domain that binds that binding pocket. And then the GRB2 also has an SH3 domain, which allows for the SOS protein called RAS activating protein in the image to bind with its PXXP motif. So the GRB2 protein has both the SH2 and SH3 domain, and then the SOS protein has the PXXP motif. And then once the SOS protein binds the GRB2 and gets activated, it acts as a RAS-GEF, which then activates RAS. So the RAS protein is anchored in the cell membrane, and in its inactive form, it's bound to GDP. And when SOS is activated, it acts as a RAS-GEF. So a GEF is a guanine exchange factor. And so when it's activated, it causes RAS to lose affinity for GDP so that GTP can go into the RAS binding pocket, which then activates the RAS protein. Any questions on this part? Yes. It's the PXSP motif would be like right over here. Right, so the SH2 domain is here, then it has the SH3 domain over here, and then that's where the PXXP motif binds.
So connected there.
Yes. Sorry. Good question. Any other questions? Yes. It's, oh gosh, now I forget what it's stands for. I think it's like a proline something, so I think it's just a bunch of like a sequence of amino acids that is recognized by the SH3 domain. Okay. Okay, so now the activated RAS protein bound to GTP goes and continues to transduce the signal throughout the pathway, and it activates a kinase cascade called the MAP kinase cascade, which is made up of 3 kinases. The first kinase that gets activated is MAP kinase, and then that when it's activated, activates MAP kinase, which then activates MAP kinase. And then this last MAP kinase goes and phosphorylates downstream targets to then result in whatever the signal required. And then another important thing to note is that a RAS gap is needed, because if there's no RAS gap to bind the activated RAS protein, then the signal would keep being transduced and it wouldn't shut off. So the RAS gap, again, is used to bind to RAS to cause it to activate its activity, which is to hydrolyze GTP to GDP. Yes. Any questions? Okay. Now we're on to the RTK PI3 kinase AKT pathway. So the purpose of this pathway is the survival of the cell. So the end result of the pathway is survival, and apoptosis therefore is the cell's default. So basically the cell, if without this pathway, basically automatically dies, and so your cells basically need to keep telling each other to stay alive, which is kind of funny. And this important pathway is a target for a lot of cancer research, because basically if you want cells to die like your cancer cells, then you would want to inhibit this survival pathway. Okay, so we're gonna break this pathway down into multiple different slides. So how does it start? The survival signal will bind to an RTK receptor, and then of course dimerization happens, cross-phosphorylation, and then the creation of the tyrosine, the phosphotyrosine binding pocket, and this causes the activated PI3 kinase. No, sorry, this causes PI3 kinase to bind to the phosphotyrosine, which then activates the PI3 kinase, and then this kinase phosphorylates an inositol phospholipid. So the inositol phospholipid is basically, is anchored to the cell membrane, and in its 2-phosphate state, it is inactive, and then once it gets phosphorylated by the PI3 kinase, it's activated. And it's not a protein, it's a phospholipid, and yes. Go ahead. No, Yes, it binds to it just binds to the phosphotyrosine, with its SH2 domain. And then that causes it to undergo a conformational change, which then causes it to get activated. And then the activated PI3 kinase phosphorylates the inositol phospholipid so that it has the 3-phosphate state. Okay. Now that the inositol phospholipid has the 3 phosphates, it can go and bind the PIP. It can go and bind protein kinase 1. And the protein kinase 1, in order to bind the PIP3, has a pH domain. So this is the pH domain over there. And the AKT, which is another protein, also binds to a PIP3 with its pH domain. But the AKT, sorry, it's a little confusing walking through the pathways, but the PIP3, oh my gosh. Okay. The activated inositol phospholipid basically is used to both activate the protein kinase 1 and the AKT. So the AKT is what's eventually going to go and inhibit apoptosis. But before it can be fully activated, it needs to be activated by 2 separate kinases. So it needs to be activated by protein kinase 1, which is activated by the PIP3. And then it also needs to be activated by the protein kinase 2. And so these 2 kinases need to phosphorylate AKT in order to fully activate it so the signal can keep going. So it first gets phosphorylated by protein kinase 1, which then allows for protein kinase 2 to phosphorylate it. And yeah, yes. Protein kinase 2 is not always active, but in order for it to be able to phosphorylate AKT, the AKT needs to, so in order for it to have affinity for the AKT needs to be first phosphorylated by the protein kinase 1. Does that make, yes. I think I usually just call it protein kinase 1 and 2. Well, it's just named differently. Oh, okay. I'm sure either works. But I can look into that later. Yeah. Okay, any more questions? Okay. Okay, so now that you have the activated AKT, it dissociates from the PIP3, bound to the cell membrane. And the reason why it dissociates is, again, because of conformational change after it's fully activated. So this AKT can then go and phosphorylate BAD, which is a protein that sequesters BCL2, which is what goes and inhibits apoptosis. So BAD and BCL2 are bound together, and then once AKT is activated, it phosphorylates BAD, which then releases BCL2, which then goes and prevents apoptosis. All good? Okay. Okay, JAK-STAT signaling pathway. The purpose of this pathway is for cytokine signaling, cell division, and for milk protein production, which is the example that we're giving here. The JAK receptor, one thing that's very important to note is that the JAK receptor is not an RTK. This is a receptor, but it does not have a kinase domain. But instead, the kinase subunits that it has kinase subunits that associate with it. So over here you have like the two like receptors, and then it is in quaternary structure with JAK1 and JAK2, which are kinases that have kinase domains, but it's not an RTK because the receptor itself does not have the kinase activity. It's the subunits that are bound to it in the inside of the cell. So this is like another like domain versus like subunit example that is important to note. So JAK stands for just another kinase, and they are non-receptor tyrosine kinases, and they're similar to RTKs, but not part of the RTK. Sorry, I'm just restating what I said. Yeah. Okay, so even though that it's not an RTK, it does function very similarly to one. So when prolactin, which is the signaling molecule, binds to two of the prolactin receptor JAK subunit complexes, dimerization occurs, and this dimerization causes the activation of the JAK1 and JAK2, which then causes them to cross-phosphorylate each other with their kinase domains. And then once the JAKs cross-phosphorylate, they then phosphorylate their respective receptors. And then this phosphorylation of the receptors provides binding sites for STAT5. And so therefore STAT5 has SH2 domains. And once STAT5 binds to the phosphotyrosine domains, they will then release from the receptor and then dimerize and then go and act as a transcription factor for milk protein genes. Yeah. Any questions? So it's important to note that all proteins that bind to phosphotyrosine domains have the SH2 domain. So the ones we've talked about so far are STAT, GRB2, and is there another one? I forget. Oh yeah, PIP3. Okay. Now on to the G protein coupled receptor and G protein signaling. The GPCRs are made up of a 7 transmembrane receptor. So it has an extracellular portion where the signal binds, and then it has multiple folds in the cell membrane, and then it has the intercellular portion where the G proteins are. The G proteins on the inside of the cell that bind the GPCR, they're made up of 3 subunits. You have the alpha subunit, the beta subunit, and then the gamma subunit. And then the only GTPase is the G alpha subunit. Okay, so the way that the GPCR protein signaling works is that a signal binds the extracellular portion of the GPCR, and then this causes a conformational change in the G protein coupled receptor. And this conformational change causes G alpha, which is bound to GTP, to lose its affinity for GDP so that GTP can bind to G alpha. And once GTP binds G alpha, it gets activated,
And the activation of G alpha causes it to dissociate from the receptor, as well as dissociate from the G beta and G gamma subunits. And then the G alpha can go and start a signal transduction cascade. Once it's done the signal transduction cascade, it can hydrolyze the GTP to GDP to inactivate it with a protein called RGS. So something important to note in this pathway is that the GPCR acts as a GEF because it causes G alpha to lose its affinity for GDP, so that GTP can diffuse in the binding pocket, and then the protein RGS acts as a gap because it causes G alpha to hydrolyze GTP so that it can go back to its inactive state. And then another thing to note is that the G gamma and G beta and G gamma complexes are permanently bound together, and they go and start their own signal transduction cascade separate from the G alpha. Okay, any questions before I move on to the next topic? All right, microtubules, structure and function, topic 6. So microtubules are very important. They are a component of the cytoskeleton and function in resisting cellular compression. They pull replicated chromosomes apart during cell division, and they provide a track for movement of vesicles and cargo throughout the cell. And they're also important for cellular structures such as flagella, cilia, and centrioles. So in research, there is a lot of visualization of microtubules in order to study cell function, and the way that microtubules are visualized usually is through GFP. And basically GFP, how it works is it's a protein, and its gene is added downstream of the promoter for tubulin. So this says favorite protein, but it's tubulin. So whenever tubulin is expressed, this fluorescent protein is expressed with it. And so wherever you see the tubulin in the cell, you'll have a fluorescent signal bound to it, which then can be viewed with a microscope in IF and immunofluorescence. That's that. Okay, so microtubule structure. Microtubules are polymers made up of tubulin monomers, which are stacked end to end, and they are hollow tubes made up of 13 protofilaments. So you have the different tubulins that are bound together, and then 13 of these filaments make up one microtubule. And the tubulin is a heterodimer made up of an alpha subunit and a beta subunit. And while both subunits can bind GTP, only the beta subunit can act as a GTPase and hydrolyze GTP to GTP. Microtubules also have a plus end and a minus end. And the minus end has the exposed alpha subunit of the tubulin, which is the non-GTPase, and then the plus end of the microtubule has the exposed beta subunit. And tubulin can be added on both the plus end or the minus end, but its addition is typically faster at the plus end, also because the minus end is usually embedded in the microtubule organizing center, which we'll talk about later. Okay, so microtubules have this thing called dynamic instability, which is very important because it allows for microtubules to polymerize and depolymerize based on what the cell needs, if it needs to divide or if it needs to bring new cargo to different places. An important thing to know about microtubules when they polymerize is that tubulin can only be added to a polymerizing microtubule when it's in the GTP bound state. In order for GTP tubulin to continue getting added to the plus end, a GTP cap needs to be present. So basically, GTP tubulin is added basically at a rate quickly enough to cause this GTP cap to form, because basically what's happening along the length of the microtubule is the GTP, the tubulin that was once bound to GTP is hydrolyzing its GTP to GTP. And if the GTP cap is not present, then depolymerization occurs because GTP bound tubulin is what causes the microtubules to stay together. Yes. And then the depolymerization is usually called like the catastrophe event. Yeah. I can go more into that if people want. How do people feel about dynamic instability? Yeah. Oh yeah. Sorry. I did not explain these two. So the two main scenarios of microtubule dynamics. So basically this is saying if the hydrolysis of GTP is occurring faster than the tubulin is getting added, that means that the hydrolysis catches up and basically the GTP cap is no longer there. So then you're going to get depolymerization. But if GTP hydrolysis is slower than the addition, that means you're going to get more GTP tubulin added at a faster rate than the hydrolysis is occurring down the microtubule. So you're going to get polymerization. So this just basically means that the you have a GTP cap because the tubulin addition is faster than the hydrolysis. Yeah. That was supposed to be depolymerization. Sorry, thanks for catching that. Okay, microtubule organizing centers. These are important in the cell because microtubules need to basically nucleate from the microtubule organizing center, because in the cell you do have GTP tubulin, but not in concentrations that are high enough for nucleation of microtubules to happen from nothing. And so basically in the cell you have two different possible microtubule organizing centers. You have basal bodies, which are the organizing centers for cilia and flagella. And they have structures similar to centrioles in order for nucleation to occur. And then there are also centrosomes in non-dividing cells and during spindle assemblies that act as microtubule organizing centers. And centrosomes, they have centrioles in the center, but they don't act as like the site for nucleation. The site for nucleation is actually the gamma tubulin ring complexes that surround the centrosome. Okay, so now we're going to talk about microtubule assembly in vitro. So this is where pure tubulin GTP is added to a test tube, and microtubule polymerization and depolymerization is assessed throughout time. When you're setting microtubule assembly in vitro, you can add as much tubulin GTP as you want. So you can add it in a concentration that allows for the nucleation of microtubules to happen without a microtubule organizing center. And so then we basically get this graph plotted with time on the x-axis and absorbance on the y-axis. And when we look at microtubule assembly and disassembly throughout time in the test tube, we see 3 different distinct phases. We see a lag phase, an elongation phase, and a plateau.
Technically speaking, shouldn't there also be like a de-escalation phase? I mean, it's not shown on here, it wasn't shown on any of the slides. But
Mhm.
realistically, it will happen.
Yeah, exactly.
So, flipping one.
Yeah, exactly. So if basically sorry, what's your name?
James.
James. What James said is that in a test tube, if more time was permitted, you would basically see a drop in the graph because you have a finite amount of GTP bound tubulin. So once all your tubulin has hydrolyzed as GTP, you get microtubule depolymerization, and then you're back to zero absorbance. Okay, so now we're going to go into the different phases. We have lag phase, and this lag phase basically occurs because you are creating microtubules from nothing in your tube. So it takes some time for like the microtubules to get started from scratch. And you have different orders of microtubule structures in lag phase. So you start with the free tubulin, and then you get tubulin dimers, and then oligomers, and then the protofilaments, and then you get the elongating microtubule. And then the more free tubulin that you have in your test tube, the shorter the lag phase, because you get more interactions between the GTP bound tubulin. And then this question, what if you add the seeds or like had microtubule organizing centers in your test tube? What would happen is that the lag phase would shorten a lot, and you would basically only have an elongation and plateau, and you would skip this lag phase because you don't need to create things, the microtubules from nothing. Okay, then you have your elongation phase, and this is basically just rapid addition of the GTP bound tubulin to the plus ends. And yeah, so you have just rapid elongation, you have your GTP cap, and then you have your plateau phase, which is basically when the rate of microtubule assembly and disassembly is equal. And sorry. Sorry, this is where James is going into, which is where the plateau phase, once all of the GTP bound tubulin is hydrolyzed, it will be bound to GDP, so then depolymerization occurs. But basically this plateau phase is where microtubule polymerization and depolymerization is happening at the same rate. Yep. Okay, so different drugs that affect microtubules. You have Taxol, which stabilizes microtubules and prevents depolymerization. You have colchicine, which leads to depolymerization, as well as vinblastine, which also leads to depolymerization. And I think just like for the exam, know how to like apply them. So like Taxol would be a good drug if you're trying to prevent or if you're trying to like prevent cancer cells from continuously dividing, because if you prevent microtubules from depolymerizing, then they can't form spindles for cell division. Okay, now we're going to get into motors. So motors are ATPases that basically move along the microtubules, and they help carry like cargo proteins around the cell. There are 2 different types of motors, you have dyneins and kinesins. Dynein is minus undirected and kinesins are typically plus undirected, but there is one minus undirected kinesin. So this is basically, did everybody watch like the 17.1 movie where it shows the, yes, okay. Yeah, so basically this is just talking through that movie, which basically shows how ATP binding and hydrolysis of the motors help the motors walk along the microtubules. When ATP binds to the motors, it causes the motors to have better affinity for the microtubules. And then once that the binding of the motors to the microtubules causes the neck linker region to swing forward, and then hydrolysis of ATP to ADP, then basically my goodness, now I'm confusing myself. Okay, sorry. I'm confusing myself, but basically the binding of ATP and hydrolysis ATP causes the neck linker regions to swing forward and then bind the microtubule head. But I would just rewatch the video because I think it's a little bit more helpful than like reading through the different steps. And that's just on Canvas. The 17.1, I think it's called, right? Okay. Cell cycle.
Okay, so moving into the last topic.
Nuisance.
Your entire thing, they were a large part of the topic there.
The slides.
I think I must have added it later. I'll go through them.
Okay,
I can go through them later.
sounds good. Yeah, well, I'm sorry, we'll cover that at the end. That was just a little slip up. Okay, so starting into an overview of the cell cycle, which is the beginning of topic 7. So during G1, which is the start, it's the first gap/growth phase of the cell cycle. During this stage, the cell is preparing for DNA replication if it receives a growth factor signal. So it has to make sure it has enough nutrients, cell machinery, no mutations in the DNA. During G1, if everything's looking good, it meets all of the requirements that it has to, it'll commit to the cell cycle at the restriction point. And that is the end. It's near the end of the phase, but once the cell hits the restriction point, there's still a little bit more that has to be done during G1 in order to prepare the cell for S phase, such as synthesizing proteins needed for S phase. G0 is a non-dividing state. The cell could have just gone through a cell cycle and is waiting for a growth factor to start the cycle again. So G0 doesn't mean it's dead. S phase is synthesis phase or DNA replication. This also has an end of phase checkpoint. And then G2 is the second gap/growth phase, where the cell prepares for chromosome segregation, which will take place during the M phase. This also has an end of phase checkpoint. And then we have M phase, which follows G2, and this consists of prophase, prometaphase, metaphase, anaphase, and telophase, which we'll kind of go into specifics a little bit more later on. And then there's also a very important metaphase checkpoint. So as you can see, there's a lot of checkpoints that happen throughout the cell cycle, where the cell kind of puts on the brakes, makes sure that everything looks good before it goes ahead and just continues to divide. Yeah. All right, so going into a little more detail about G1. As I said before, cells are still in G1 after the restriction point, which is about where that red arrow is pointing. So there's still a good section of time where the cell is not yet synthesizing its DNA, but it's committed to the cell cycle. So G1 is the beginning of the cell cycle, entry point. Most eukaryotic cells need to receive a growth signal, such as a growth factor hormone. When they receive this growth signal, it just means that they can start thinking about dividing. So receiving that signal doesn't at all mean that they're going to divide. They've just received a signal that tells them, okay, it's time to start looking and seeing if, checking for those requirements and seeing if the cell can proceed through the cell cycle. So these requirements include checking for DNA damage, correct size, energy and nutrient content, such as carbon and nitrogen, which are all really important for example, nucleotide formation. And I didn't mention this, but the the growth factor signal is usually transduced through the RTK RAS MAPK pathway. And then if these conditions are met, cells will pass through the restriction point, which is the point of no return. If the cell passes through the restriction point, it must either complete cell division or die. And the restriction point in yeast is called start. Yeah, so finish the cell cycle after restriction point or undergo apoptosis, aka cell death. All right, and then we have S phase, which comes after G1. We define S phase as when cells replicate their DNA. The S phase checkpoint is when cells can temporarily halt DNA replication if an error is detected, and then resume DNA replication or perform programmed cell death or apoptosis. So for example, if the cell detects DNA, good question.
So is there a checkpoint after our checkpoint that you want to ask?
So the restriction point is the main checkpoint in G1,
Yeah.
and then it'll continue synthesizing proteins, going through the last few steps. Then it goes into S phase, and then the S phase checkpoint would be the next checkpoint after that.
So S phase checkpoint would be after G2.
It would be during S. Yeah.
Oh,
Yeah. So it's like if during S phase, when the cell is replicating its DNA, it detects DNA damage of some kind, it can initiate a response that can either repair that DNA or cause cell death.
Okay.
And an example of a regulatory mechanism that would respond to DNA damage is the P53 pathway, which I will be discussing more in detail later on, but just keep that in mind. Okay. All right, and then after DNA synthesis, S phase, we have G2, which is a second growth phase, where the cell is basically just making sure that DNA is completely replicated, it's been replicated correctly, and the cell also needs to continue to grow to make sure there's enough organelles, there's enough physical size in order for the cell to divide during M phase. So then we have another checkpoint during G2 where the cells cannot enter M phase until they have grown to the correct size and thoroughly checked their DNA. So if the cell is at a good enough size to divide and all of its DNA looks good, undamaged, it will proceed to M phase, where spindle assembly and chromosome segregation will occur. So during M phase, we have prophase, where the nuclear envelope is kind of dissolving, metaphase, where chromosomes line up in the middle, anaphase, where those chromosomes are separated, telophase, where our nuclei are fully independent of each other, so they're fully separated, and then cytokinesis, where we have two daughter cells. And then the checkpoint that takes place during M phase takes place, it's called the metaphase checkpoint, and it takes place during metaphase and prevents the cell from entering anaphase until chromosomes have proper microtubule attachment. So this is important.
Is that actually SAC?
What was that?
Single assembly checkpoints?
Yeah.
Oh,
Yeah, sorry, I just didn't specifically say that, but yeah, spindle assembly checkpoint slash metaphase checkpoint, same thing. Checking for correct attachments to chromosomes. Because if those microtubules aren't attached correctly, then you can get instead of a nice, even division, you could get all of one going to one side, etc.
actually, I
Bad for
have a
daughter cells.
question about
Sure.
the MPs. I'm gonna have to look back at the bottom set. I remember that there was like one thing in Topic 7 that was really giving me some hope. It said, name and discuss two chromosomal sequences, structures, and areas whose dominance to ensure that duplication of chromosomes are kept constant from one generation to the next. I expected that spindle assembly checklist was one of them, but I could not for the life of me figure out one second.
So I know exactly what question you're talking about. It was it was a little bit hard to piece what the two parts was, but the first one was, would you mind reading it? The question again?
Name and
Yes.
discuss two chromosomal sequences, structures, areas, and their functions whose job it is to ensure that duplication and number of chromosomes are kept constant from one generation to the next.
Okay.
We briefly discussed them this week in topic 6 and the required for DNA replication and chromosome segregation. But I looked at topic 6 like constantly, I couldn't find anything of it to be related to. My closest idea was maybe microtubules, but those don't move off there. Maybe kinesis, but kinesis aren't really chromosomes.
Okay, so you're on the right track. So the two important things there are duplication and segregation. So for segregation, you're absolutely right. You're looking for a structure on the chromosome that is important during segregation. So you're thinking where those microtubules will attach on the chromosome.
So we just talked about that.
What was that?
Cap. Is it cap part of it?
So it's where the kinetochore will attach, which is the centromere.
Central. Is it centromere or centrosome?
Centromere,
I think.
I believe.
It's centromere and spindle checkpoints.
So the question was looking for a structure on the chromosome, so it would be the centromere. But so when it says two, it's asking for correct duplication, which is separate from, so that would be like during S phase. And then segregation is during M phase.
It literally just name and discuss two chromosomal sequences, structures and areas. It's directly asking for two.
No, yeah, and the two are, one is related to duplication and one is related to segregation. I know the wording's a little. Do you mind if we come back to this
Sure.
after the review session? Sorry, I just don't want to. Okay. So going into regulatory mechanisms of the cell cycle, we have our master cell cycle regulator, which is cyclin-dependent kinase or CDK. It is a serine threonine kinase, which is just basically indicates the type of amino acid that it can phosphorylate. It's regulated by cyclin levels, so varying different types of cyclin will be expressed throughout the cell cycle. It's regulated by phosphorylation, so it'll be activated by CAC or CDK activating kinase, and it'll be deactivated by Wee1, which deactivates it by phosphorylating it. Which is a key thing to remember here. Phosphorylation doesn't necessarily mean activation. For Wee1, it would add an inhibitory phosphate like here. And then even if CDK is phosphorylated by the activating kinase, it cannot function if it's phosphorylated by Wee1. It won't be active, if that makes sense. And then there's also CDK inhibitors, which are proteins that will directly bind to CDK, such as P21. And then that also inhibits CDK's function and puts the brakes on the cell cycle. Okay, in order to be activated, CDK needs to be both phosphorylated by CAC and be bound to cyclin. You have to have both of these happening at the same time. Anytime you reach a checkpoint, the cell brakes are put on by inhibiting or deactivating CDK. So inhibiting or deactivating via phosphorylation or binding by inhibitory protein. CDKs target different substrates throughout the cell cycle, depending on the cycle phase it's in. And depending on the cell cycle phase, a different cyclin will be expressed that causes that substrate specificity change depending on the cell cycle. And again, CDK cannot work as a kinase unless it is bound to cyclin. Exactly. Yeah. Off the top of my head, Rosella, would you know that? Okay, so the question was, so CDK adds a phosphate to serines and threonines, but which amino acid is on CDK that is phosphorylated? Does that, do we need to? Okay, that's a great question, but you do not need to know that for the exam, so I wouldn't worry about it. If you're curious though, definitely feel free to ask Dr. Francis.
I'm assuming that's from what I read in the earlier questions that the reason why it's so breaks are there is because CDK is heavily connected to basically DNA creation. Like it's connected to a lot of those sequences. So I'm assuming the reason why it's so heavily regulated and so DNA replication isn't occurring when it's really damaged or unchecked.
Yeah, exactly. That's how they posit
Yeah.
is by halting CDK activity because it's there throughout the cell cycle, but it's deactivated if they need to take a pause and fix things. So going into more detail about cyclins, they regulate CDK function by activating kinase activity. They have to be bound to CDK in order for CDK to be active. And by binding to CDK, they also influence CDK binding specificity. So this means that they alter, because of conformational change in CDK that changes what it can phosphorylate to make sure that it's targeting the correct proteins during the correct stage of the cell cycle. Cyclin itself is only regulated by production, which takes place through transcription, translation, and destruction, which occurs through ubiquitin-mediated proteolysis. So cells produce the cyclin they need based on the cell cycle phase. It's basically just it basically relies on the concentration of each type of cyclin. So for example, you'll have G2 cyclins, the green one, present during G2 phase that will bind to CDK during G2 phase and carry out important processes during G2, such as growth, checking for DNA damage, things like that. CDK and cyclin complex. So CDK alone has an activation loop which phosphorylates and dephosphorylates other proteins. It's tucked inside the CDK and not available to bind to anything. But when cyclin comes along and binds, CDK undergoes a conformational change which exposes that activation loop. And now the CDK is physically able to phosphorylate other proteins. But at this point, just because that region is exposed, that doesn't mean it's fully active and will not yet phosphorylate proteins. So at that point, CAC needs to come along to add a phosphate to CDK's activation loop, which allows CDK to phosphorylate its target proteins / substrates. And then there's also CDKIs or CDK inhibitors, which block target proteins or prevent CDK from binding in some way. Again, cyclins are phase specific, so that CDK can regulate particular proteins in particular phases of the cell cycle, depending on which cyclin type is present at that time. And this is caused by the change in substrate specificity, depending on which cyclin CDK is bound to. CDK is regulated by phosphorylated kinases. CDK must be phosphorylated by CDK activating kinase in order to function as a CDK. And it cannot have a phosphate from Wee1 kinase in order to be active. So when Wee1 has phosphorylated CDK, it's inactive. Phosphatases will dephosphorylate CDK, which can either take off the inhibitory phosphate put on by Wee1, which would help it be active, or take off the activating phosphate put on by CAC, which would cause CDK to be inactive. And CDK inhibitors are typically seen in G1 regulation or response to DNA damage. So again, an example of a CDK inhibitor that would be present in response to DNA damage is P21. Again, it's target for molecular breaks, stopping the cell cycle if need be. And I mentioned P21. Okay, so going further into how P53, P21 works. So if the cell detects DNA damage, P53 is activated through phosphorylation. P53 is a transcription factor and is a tumor suppressor because it codes for a protein that inhibits CDK, which stops the cell cycle. So when P53 is active in response to DNA damage, it activates transcription of P21. And when P21 is translated, it binds to CDK cyclin complexes, which inhibits the activity of CDK, which puts a brake on the cell cycle to give the cell time to either repair damage or undergo apoptosis or programmed cell death. And P53 is heavily regulated or degraded, or if inactive. So why is P53 a tumor suppressor? That's because it codes for a protein P21 that inhibits CDK, which puts a brake on the cell cycle. So if you have overproliferative cells that should not be going through the cell cycle, and P53 is activated and leads to transcription of P21, the cell cycle will halt, stopping those overproliferative cells. And if P53 is no longer available, we lose that regulatory mechanism, and the cell, that is one regulatory mechanism that would be lost. And the cell is free to continue dividing even if there is DNA damage, which would be very negative for the cell. All right, so specifically mechanisms of control and regulation in the G1 phase. So before cells enter the cell cycle, they must receive signals such as growth factors so that they can start thinking.
So is P53 active in any particular phase, or is it just stop around the whole cell cycle?
So P53 can kind of be around, it's produced and degraded, but it will only be activated if the cell senses DNA damage. So then that leads to phosphorylation of P53, and then that leads to transcription of P21. Okay, so they have to receive growth factor signals so they can start thinking about dividing. They check for DNA damage, check for early G1 cyclin, which is cyclin D, and they check for size, nutrients, and energy. So this is all taking place before the restriction point. And they make sure the pre-replication complex formed, which is also done before the restriction point during G1. So the pre-RC is a protein complex at the origins of replication and is required for DNA replication. It begins when activated CDK levels are low in G1 before the restriction point, And why is that? The cell just kind of has to be mm. Okay, because CDK presence throughout the rest of the cell cycle inhibits the pre-RC to make sure that the cell isn't replicating its DNA more than once. Okay. So during the formation of the pre-RC, we have recruitment of various proteins. We have ORC first, which is DNA binding protein. It binds to the origin of replication. It is an ATPase. ORC then recruits CDC6, and then CDC6 recruits CDT1, and then the helicase, MCM comes along and binds over here. And then once this is loaded, the CDT1 can disassociate. So as the helicase, the MCM will be the protein that peels apart the DNA during replication. So once that pre-RC is present, that's known as origin licensing. And that takes place before the restriction point. And then the next step after the restriction point, the cell starts to recruit more proteins to form the pre-initiation complex, and that requires active CDK.
So just like I have the timeline in my head, right?
Mhm.
It's the CDK cycling complex defines the phosphorylase RB if these E2F is free to move, and then the pre-IC is formed. Because that is the restriction, right? So that happening, and then the pre-IC is formed after. And then
Yes,
the origin firing.
yes.
Okay.
Yeah. Nope, sorry.
I was just gonna ask, should we be familiar with the individual components of the PRC, the CBT and the ORC?
Maybe I wouldn't say you have to know like which one's a helicase necessarily, but maybe have an idea of what the pre-RC, what the difference between the pre-RC and what the pre-IC is.
Well, like, should we, do you think we have to know like the specific names of the?
If you have space on your, I mean, I can't say for sure, that's probably not, that's in relation to everything else, it's a smaller detail, so
Yeah.
probably not, but I can't say for certain. So if you have space, include it, so you don't have to like memorize it, you can just write it down and then not think about it. But most likely not, I would say. Okay, so during G1, CDKs get fully activated. If cyclin CDK has inhibitory and activating phosphates, so this would be the inhibitory phosphate put on by WEE1. Even though it has this activating phosphate and it's bound to cyclin, it's inactive if this phosphate is present. So we have to have a phosphatase cleave this inhibitory phosphate, which now with this gone, CDK cyclin complexes can be active.
That's.
Hmm?
That's a picture.
Yeah. Okay, so now that CDKs are activated, getting into the restriction point, RB is a protein that when bound to E2F, which is a transcription factor, E2F is deactivated. So once we have activated CDKs, those can phosphorylate RB, which causes it to let go of E2F. And then E2F can promote transcription of proteins required for S phase. And then that leads to the formation of the pre-IC complex and the recruitment of DNA polymerase. And then the complex becomes a replosome. So this is origin firing into S phase, and the cell has past the restriction point, aka the point of no return, so the cell must either completely divide or die. Yeah, so I guess the picture, RB is holding on to E2F. You want RB to let go of E2F in order for the cell cycle to take place.
So after origin firing is done, it's going to escalate.
Yes, so origin firing is when, is that when duplication begins, officially begins? Okay, yeah, so S phase starts officially when duplication begins. And that is origin like firing.
Okay.
Yeah. Okay. Does anyone have any questions about anything? So the pre-IC is, the pre-RC is formed early in G1 when there's low CDK levels. And that's the only time that it can form, because during the rest of the cycle, CDK is pretty much active. And then that happens before the restriction point. And then the pre-IC forms after the restriction point, adding more proteins on, which leads to origin firing. Sorry, was that? Yeah, so here, I'll go back. Okay, so this is the pre-RC where the helicase, everything is added. Oh, there isn't a diagram of the pre-IC. Okay, well, the main difference is that pre-IC complex leads to recruitment of DNA polymerase, which is what replicates the DNA. And then that complex becomes a replosome. Does that help? Okay. Anything else?
Wasn't the place? Remember last time? That I think it's a while for the active offloader.
So I think we can send them out tonight. Yeah, we'll send them right after. Sorry. Ah. Yeah, I added a slide later too, and it didn't.
Yeah, I don't know why. Okay. So these are just the different kinesins. Kinesin 5 is a tetramer, so it has 4 different components, and it has both heads and tails on either end. So when it's active, it can bind to overlapping microtubules. This is important for cell division so that the different poles get separated. Then kinesin 1 and 3 help to bring cargo proteins up and down the microtubule. Kinesin 1 is a dimer, and then kinesin 3 is a monomer. But when they're active, they both have 2 subunits, and the cargo is bound to the tail end, and then the head is bound to the microtubules. And then kinesin 13 destabilizes plus ends of microtubules and just like slides across. And then there is a minus end kinesin, and I think that that's kinesin 14. Yeah, that's the extent to which you need to know the kinesins. Thank you guys. We can answer any questions now if you guys haven't.